Okay, thanks Eric and Eric for the invitation to talk today. So yeah, my lab studies these chemosynthetic iron oxidizing bacteria. And I'm the one up here standing and presenting today, but this is really the work of Gerald Murray, who just got his PhD in Kirsten Kusel's lab at the University of Jena, and Roman Barco, who's actually an NSF postdoc who splits his time between my lab and Ken Nielsen's lab at USC. And I'd also like to thank NASA Astrobiology for funding and NSF. And in case any of you were wondering what that little symbol, oh shoot, I knew this was going to happen. That little symbol there, that's the alchemist sign for iron, which is what we study. So why iron? First of all, it's probably the most abundant chemosynthetic element on Earth and probably on any other rocky planet that we know about. Well, maybe I shouldn't say that, but being at an astrobiology meeting, but anyway, I'll say it anyway, because I'm a microbiologist and I can say stupid things about geology and planets. So another thing is biosignature. So I'm sure a number of you have seen this paper that recently came out in Nature where they were claiming for these iron oxide biosignatures in 3.8 or older billion-year-old rocks or older from the Hudson Bay. And, you know, very interesting. I study modern systems. I've never actually gone out in the field and collected a rock or looked at a rock slide under the microscope. But when I look at these types of structures, you know, they are somewhat consistent. They certainly are not totally consistent with anything we see in the modern world, but, you know, after 4 billion years, who knows? So I guess I'm a hopeful agnostic in that this work will really be borne out. But just to show that you can find microfossils that are very well preserved. In this slide here is a 170 million year old Earth crust where you see all these little filamentous structures that are about 1 or 2 microns in diameter. This is about a 3-week-old culture of an iron oxidizing bacteria that was grown in the lab. And you can see there's a lot of similarity in those structures. And then when you look at the fine detail, it becomes pretty clear that some of these types of structures clearly must have been formed by microbes. How closely related they are to the microbes we have today, that's another question. So we definitely see biogenic structures in nature that these organisms form, and they can be small and large. So on the left here is a intact microbial mat that we did a 3-dimensional imaging of with a confocal microscope, and it was formed by this sheath-forming bacterium here. What's really cool about this is that 50 micrometer or so layer at the leading edge of that mat is where all these sheath-forming bacteria are, and all this red material is just the empty sheaths that they leave behind. So they really are geoengineers or ecological engineers. They structure their environments and produce massive amounts of this amorphous ferrihydrite, which is quite a reactive mineral. To go to the other scale, this is the Golden Tower, which we discovered a couple of years ago while doing submersible dives at the Hiroshima event site in the Mariana. That little bar down there is 10 centimeters, so this structure is nearly 10 meters tall, and it seemed, as far as we could tell, it was composed primarily of bacteria that looked like this, that form these long stalks. So this is formed over a diffuse vent flow where there was iron coming out, and it just slowly built up over time, as far as we could tell, and formed this massive structure. These bacteria are interesting because they undergo very little grazing and things like that by protozoa or shrimp. So they don't undergo the same kinds of pressures that, for example, sulfur oxidizing mats do. So they have the potential to really accumulate. So what's going on? How are these bacteria actually oxidizing iron? And that's the primary topic I want to discuss today. So this is a model that we came up with, published last year, which is based on both comparative genomics and some whole cell proteomics we did with an organism we've been studying in the lab for a while, Mary profundus ferrooxydans. So this is an example of extracellular electron transfer, because the cells, so they oxidize iron on the outer membrane, because again, they have the problem in neutral pH, as soon as you oxidize iron, you immediately precipitate a ferrihydrite mineral. If you do it inside the cell, you're going to fill your cell with insoluble iron oxides. So we believe this process should occur at the outer membrane. So there you have to get electrons from the outer membrane across the periplasm to the inner membrane, to the electron transport chain. And so what we found in all the iron oxidizers, both from freshwater and marine systems that we've looked at, is this CYC2 homolog, which we call CYC2PV1. And we believe this could be the initial iron oxidase. This is it's a porin cytochrome C complex, and then electrons would, so that would be the initial electron acceptor, and then electrons get transferred across the periplasm through, we've found some other cytochromes. It could be potential shuttles, whether these are shuttles or could form a wire, we don't really know. And then there's an electron transport chain, which has the different components. Interesting aspects of this are this alternative complex 3, which we quite commonly find, it's a molybdenum oxidoreductase system. And then most of these organisms use the cytochrome CBB3 type terminal oxidase, which is a high affinity iron oxidase. That makes sense because we think these organisms are generally microaerophilic. So that's what we've known, and most of the organisms we've isolated have been obligate iron oxidizers, I should say. But recently, or actually it's not that recent now, but a few years ago I isolated this new organism, which we're just in the process of publishing on and of calling it Ghiorsea bivora. It represents a new genus in this group of Zetaproteobacteria. The Zetaproteobacteria are almost exclusively iron oxidizers that live in marine environments. So I isolated one strain from the tag vent site in the mid-Atlantic ridge and another one from the snail vents in the Marianas, so on the opposite sides of the world at least. And it's kind of a nondescript organism in terms of a lot of the other iron oxidizers. It's a little rod, and it forms these very amorphous particulate iron oxides. It doesn't make any of those structures that I was showing earlier. But what's really cool about it is that it can grow not only on iron, but also on hydrogen. And so those organisms were isolated on, or enriched and isolated on zero-valent iron, which is both a source of iron, and we use it as that, but you also have to be careful because it can be a source of hydrogen as well. And it turned out this organism grew really well in the enrichments, and part of the reason was because it was growing both on the iron and the hydrogen. And when we separate those out and grow it on ferrous chloride or hydrogen, you can see in the squares here is the hydrogen. It grows a little bit better. And in the circles here is growing on ferrous chloride, and pretty decent cell yields and doubling times on the order of 15 to 20 hours. If you don't add either hydrogen or iron, there's no growth. So we sequenced the genomes of these two organisms and found that they did indeed have hydrogenase, the hydrogenase subunits for uptake of hydrogen. And then we compared this, so we have a bunch of single cell genomes from other Zetaproteobacteria that we've isolated, or sorry, that we haven't isolated. We've isolated the single cells and sequenced the genomes. And we found that they all belong to this single clade here, which we call Zeta-OTU9. And they were different from these other isolates that we have, which represent different clades within this group. And so that was interesting. So it seemed like it was this one clade that we have that it seems to have the ability well, it has these hydrogenases that are capable of promoting growth on hydrogen. And so then we were asked the question of, well, where are these found? And so we scraped the short read archive to look specifically for this particular group. And so we found about 50,000 projects that have been done that are non-human associated. There's about 30,000 human associated projects in there. And we found only about 4 or 5 projects where this particular OTU is found, and they were all in high iron environments. That makes us think these organisms, although they can grow on hydrogen, they may be more adapted for growing on iron. So what can we learn? So I meant to say that the beauty of having this organism is everything we'd done before was done on obligate iron oxidizers. So now we have an organism that can grow on both hydrogen and iron. We can at least do some comparative proteomics in this case. And so this was work that Roman's been doing. And so I'm showing here is the proteome map. So as I said, we sequenced the genome. It's about 99+% complete, but there are gaps in it. So these show the 13 different contigs on the outside here, which is the genome. And then all the proteins that we identified are in this inner blue ring here. And then proteins that are upregulated on hydrogen are shown in the green, and on iron in the red. So basically, we found about 42% of the proteins in the proteomics experiment, and about 100 of them were upregulated on iron, and about 150 were upregulated on hydrogen. And it's easy for me to put this slide up here and talk about it, but it took Roman several months to do this. He had to grow all these cells in triplicate batches to get really robust statistics. The challenge with these, we can't do genetics with them. And the challenge with biochemistry is getting enough biomass, and then you've got all these iron oxides around that you have to deal with. The hydrogen actually was a lot easier, of course, because we didn't have the iron around. But still, it was a technical challenge to do this. And so what did we find in terms of the comparative proteomics, in terms of what was expressed and what wasn't? So we found the hydrogenase operon on hydrogen was more highly expressed. That was good. We found that the phosphate, that there's a transport system for phosphate, which was highly expressed on iron. This is actually a nice internal control, because as these cells excrete iron oxides, those iron oxides very strongly bind phosphorus. And so the fact that they overexpress these systems for taking up phosphorus is sort of makes sense. There was also this thioredoxin reductase, which is involved, and one of the things it's involved in is probably defense against reactive oxygen species. Again, when you have iron around and oxygen, you're going to get the potential for reactive oxygen species that these organisms have to deal with. What was not differentially regulated was rubisco, their ability to fix CO2. That's not surprising, or the CBB3. The ACT system also was expressed in both cases. And the CYC2 was also expressed about equivalently. This was a little disappointing, I guess, because, of course, we were hoping to see this upregulated when they were growing on iron and downregulated on hydrogen. So the case was true for the hydrogenase, but not for the CYC2. So I'll just come back to the model here. So, it was good that we saw CYC2 highly expressed in these, because we'd only done this in a different species before, so it's consistent with it being there. We didn't see the CYC1. We think there's a different system that they may use for shuttling electrons. So, what's going on here in terms of this? Well, I can wave my hands and say that they're probably constitutively expressing the CYC2, and the primary argument I could make for that is ecological, right? That these organisms really want to grow on iron, and so they're always ready to grow on iron. But if some hydrogen comes along, then they can adapt and take over and use that instead. So with that, I'm going to conclude. Again, these neutrophilic iron oxidizers are obligate, and this utilize extracellular electron transfer. And as I said, CYC2 seems to be constitutively expressed. And I just talked about an ecological reason for that. But another thing that we're thinking about that would be really interesting now is perhaps Ghiorsea bivora is a sentinel for the presence of hydrogen at diffuse flow vents, because we only see it in some communities. So it'd be interesting to go out and see if those are the communities where there's more hydrogen present. So, thanks.
Thank you, Dave. Time for, again, probably one question.
Hi, David. My name is Sanjoy Som. I was wondering if you could expand a little bit on the electron transport mechanism across the membrane.
A little bit. So, we think that there's these cytochromes that are probably involved in shuttling electrons from the outer membrane or from that. I mean, we're still, I mean, our model is still based around a CYC2-like protein as being that outer, that does the initial contact with Fe2 and oxidizes to Fe3, and that then those electrons are transferred. And yeah, so we don't find some of the canonical proteins that have been found, for example, in acidithiobacillus that are involved in that process. You know, we don't find necessarily the same systems that would be in some of the iron reducers, which go in the other direction. So, you know, whether this is some kind of a shuttle system where there's a soluble cytochrome that does that transfer, or whether there's actually a heme or a chain of hemes or something like that that act as a wire. These organisms have some fairly large heme groups in them, but not as large as, for example, Geobacter. We don't tend to find cytochromes that have, say, 30 or 40 hemes in them, or hemes groups in them. But some of them have 10 to 20. But you know, we're trying to sort that out. One thing I would say is I don't think there's any universal mechanism here. I think, you know, each system, each organism, maybe not each organism, but I think there's going to be quite a bit of modularity in this system.
Thank you.
Okay, thanks, Dave.